In this video, I'm going to discuss topoisomerases, which are the enzymes that regulate DNA supercoiling in cells. The job of a topoisomerase is to change the linking number of DNA. They do this by transiently breaking one or both sugar phosphate backbones of the double helix, and they do some stuff, and then they join the strands back together. Now we classify topoisomerases into two types as shown. Type 1 creates single-strand breaks transiently, and type 2 creates double-strand breaks. Now we're not going to go into great detail about the mechanisms of topoisomerases, but it is helpful to get some sense of how these enzymes work. This is a great figure that summarizes the three known mechanisms of topoisomerases. Type 1 topoisomerases are broken into two subclasses based on their mechanism. Type 1a enzymes cut one strand, then pass the other strand through the resulting gap. So in the figure you can imagine the enzyme cleaving the back strand and then passing the front strand through so that it's now behind the cut strand. Then the gap is sealed. Each time you do this, you change the linking number by 1. It turns out that type 1a topoisomerases only increase the linking number and never decrease it. So the L goes up by 1. You can think of this as introducing positive supercoiling or as removing negative supercoiling. So if W is below 0, like -4 as we saw in the previous video, type 1As can bring the writhe closer to 0 in steps of 1. Also, they only relax DNA, or bring W closer to 0. If you have positively supercoiled DNA, type 1A topoisomerases will not act on it at all because that would mean making W even more positive or less relaxed. The mechanism does not involve hydrolysis of ATP and has no energy input. To be spontaneous, it relies on the favorable process of relieving supercoiling or relaxing the DNA. And because it only goes in the one direction, that of increasing the writhe and the L, type 1A enzymes will only act on negatively supercoiled DNA. Type 1B topoisomerases also cleave only one of the strands, but rather than passing the other strand through, they hold on to the one side and let the other side freely spin or rotate around. This is a less controlled mechanism than the type 1A enzymes. The rotation just spontaneously happens to relieve supercoiling if the DNA is given a chance, so again, no ATP or other outside energy input is necessary. This mechanism can relieve positive or negative supercoiling, or bring the value of W closer to 0 from either direction. Which way L moves depends on how the DNA is supercoiled. The number of rotations is not fixed, so L will move by some number n each time a type 1b topoisomerase acts, and not necessarily the number being 1. Finally, we have type 2 topoisomerases, which cleave both strands. In this diagram, we have both strands of the double helix represented. Both strands are cleaved, and the double helix is passed through the gap. So the mechanism is similar to the type 1A enzyme in which one strand is cleaved and the other is passed through the gap, except here both strands are cleaved and two strands are passed through the gap. The type 2 enzyme changes the linking number by 2 because you are moving two strands relative to two other strands. Type 2 enzymes can resolve either negative or positive supercoiling, bringing W closer to 0 from either direction. These enzymes require energy input in the form of ATP hydrolysis. These different mechanisms are compared in the table on the next slide. So in summary, we've got both types of type 1 topoisomerase making a single strand break and type 2 making a double strand break. That's how they're defined. Neither type 1 uses ATP, but type 2 does require ATP. For type 1 enzymes, the energy for the reaction comes from the spontaneous nature of just relieving the supercoiling that exists, relieving that tension. Note that type 1A can relax only negative supercoils, whereas types 1B and type 2 can relax either kind of supercoiling. And the changes in linking number reflect the mechanisms. Type 1A increases L in steps of 1. Type 2 increases or decreases in steps of 2, whereas type 1b is a little more free, and for each strand break you could get a different change in L. So why these enzymes exist at all? Well, supercoiling is introduced during replication and transcription, as we are going to see in the upcoming classes. This supercoiling has to be controlled. So if supercoiling is allowed to accumulate, you'd end up with just a tangled mess of DNA that's very difficult to handle. And also, during cell division, if the strands are crossed or somehow tangled, that would prevent them from being separated into the daughter cells. So topoisomerases exist to resolve any tangles or catching that happens when two DNA strands can't get past each other during cell division. On this slide, I quickly want to introduce a specific type of type 2 topoisomerase called DNA gyrase. This enzyme is found only in bacteria. Now, whereas most type 2 topoisomerases remove supercoiling to bring W closer to zero, DNA gyrase is a special exception that uses the energy from ATP hydrolysis to introduce negative supercoiling, and it will actually move W away from zero to make negatively supercoiled DNA even more negatively supercoiled. This figure from your book shows the mechanism. The enzyme breaks two strands and passes the double helix through the break. You can see that we end up with a writhe of -2 after starting with the writhe of 0. We will see why DNA gyrase does this when we discuss replication. Okay, so to summarize supercoiling, we got twist, which is the coiling of single strands around each other in the double helix. We've got writhe, which is the coiling of the double helix around itself. And linking number is just twist plus the writhe, and is the total number of times the two strands wind around each other. And I introduced the three different types of topoisomerases and how they function. So in class, we're going to do some exercises with some physical objects that I hope will clarify these concepts and help supercoiling make sense.